First of all, I'd like to thank the CannMed advisory panel to give me this opportunity to share our study. So first of all, I'd like to share what was the purpose of this study. To understand the status of cannabis microbial testing regulations in all U.S. states and territories. Then second, to identify trends in microbial testing rules since 2019, and to identify a consensus set of required microbial testing rules to lower public health risks. Now, everyone knows what the federal perspective of cannabis is, but there are individuals from other countries. First, the use, sale, and possessions is illegal under federal law. It's considered a Schedule I drug under the Federal Controlled Substance Act. And also, and Schedule I drugs are considered to have no accepted medical use, a high potential for abuse, and physical and or psychological dependence. So where is cannabis legal in the United States? And this is as of January 2023. So highlighted in gold, 21 states and 2 territories have both legal adult use and medical cannabis programs. Highlighted in green, there are 16 states that have medical cannabis only, that would be 2 additional territories. So the total number of jurisdictions in the United States is 42. This slide shows the methods of our study. First, we reviewed all the current cannabis testing rules. Then we updated our microbial testing regulations for each jurisdiction, and the 2 foci we had was required microbial test for each jurisdiction and the corresponding action level for each test for each product type. And there's 4 product types: flower, concentrates, edibles, and non-edibles. And the web link where you can find all this information updated to the present is shown in the slide. So from this data set, we determined for each test the number of jurisdictions requiring each test, the number of each action level for each test, and the number of unique microbial test combinations for flower and edibles. But this afternoon, I'll only be showing you the data for the flower testing. Then we performed 2 sets of comparative analysis. First, within the 2023 data sets for testing flower and edibles, and also a comparative analysis between a 2019 database and our present 2023 database to see the direction of where microbial testing is occurring in the United States. So here are some general observations concerning the required microbial testing regulations in the U.S. The bottom line is it's a mess. There's 22 different microbial tests that are required by at least 1 jurisdiction. And they fall into 3 categories. The first category is total count tests. There's 6 of them. And I have 4 examples: total aerobic bacteria, total yeast and molds, total coliforms, and total biotolerant gram-negative bacteria. And then there are 4 tests where it's specific genuses, any species. So these first 2 types of tests, I'm not a proponent of requiring this testing because an enumeration of these 2 kinds of tests provides me with no information whether there's any pathogenic microorganisms in my sample, and I might get a failure in my sample. Then the last type are there's 12 specific genus species tests, and in the literature, in the clinical literature, I have 5 examples here. We have the 4 pathogenic Aspergillus species, that's Aspergillus flavus, fumigatus, niger, and terreus. and shigotoxin-producing E. coli, one of 6 pathotypes, where this one is the most pathogenic. Now, interestingly, of all the jurisdictions, the number of microbial tests required was a wide range. One jurisdiction has just 1 test required, and a few states as many as 10 tests. So an example of the sole state in the United States that only requires 1 state is Arkansas, is the absence of total coliforms. But anyone can Google this, and not all coliforms are pathogenic. So you might get a fail, but are there any pathogenic coliforms in the sample? I don't know. And then Connecticut is over the top, where they have the 6 specific pathogens that I've described, including also Salmonella species, but also 4 total count tests. Now, there's no consensus even in the action thresholds, and the widest range for the total count test is the total yeast and mold. On the low end is, it's 3 orders of magnitude. On the low end is 1,000 colony-forming units per gram of sample, and on the high end is 10 to the 5th. And for all the genus and the genus species test, it's always non-detect. Now, this slide describes the status of the microbial testing rules for flowers in the start of this year. So if you focus on the second row, I've already mentioned the 42 jurisdictions that have legal programs, but 3 of the jurisdictions, being 3 of the territories, have no testing rules to date. Now, there's actually 41 sets of microbial testing rules because 2 states, Arizona and New York, have separate testing rules for adult and medical use. So what we have is 13 of these states share 3 common testing rule sets. We have what's commonly known as the California model, which are the 6 pathogens. That's Salmonella species, shigotoxin-producing E. coli, and the 4 pathogenic Aspergillus species. 3 states have tacked on total yeast and mold, and then 6 states still utilize the archaic American herbal pharmacopoeia model that came out in 2014, which are 4 total count tests, total aerobic, total yeast and mold, total coliforms, biotolerant, gram-negative bacteria, and 1 nonsensical rule: pathogenic E. coli. Please correct me if I'm wrong, but I have not ever found a single test utilizing any technology that can detect all 6 pathotypes of E. coli. And so I've asked lab directors, what do you do? Oh, we test for shigotoxin-producing E. coli. That's what they do. So what we have, the bottom line is shown in the lower right, is that there's some chaos here still. There's 29 unique test combinations in the United States, and so there's still quite a bit of chaos. Now what we have is I'd like to share with you the trends over the last 3 years in terms of the total count tests and the specific human pathogens. And this slide is the total count test. And the bottom line is that there's a trend towards decrease in the fraction of the jurisdictions asking for this test. So the top line is the total aerobic bacteria and total microbial count, whereby over the 3-year period, the fraction of jurisdictions has decreased from 67% to 44%, and that being a change of 23%. The total yeast and mold, they're hanging on for dear life, whereby it's decreased from 74 to 63, a decrease in 11%. And then the third line, total coliforms, they're hanging on to that one too, but it's a lower percentage of the fraction as of 2023. It's 37%, a decrease in 4%. And the biotolerant gram-negative bacteria have decreased the most, down to 32% of the jurisdictions, was a decrease of almost 40%. Now what we have now is the specific pathogen tests, and this shows a trending, in my mind, the correct direction towards consensus as a beginning point. So we have the Salmonella species, it was already 85%, but additional states have seen the light, and now all jurisdictions except 2 require Salmonella testing. And then for the pathogenic E. coli, that nonsensical rule has now decreased to 27% of the jurisdictions, a decrease in 17%. And this is an indication that regulators are being educated that this is a rule that can't be met. And so until a method developer develops a test that can detect all 6 pathotypes, I feel that the most pathogenic one should be tested for, that being Shiga-toxin-producing E. coli, and there has been a dramatic increase to 44% of the jurisdictions, which was an increase in 29%. And sorry for the typo, the Aspergillus pathogens have increased also dramatically to over half, 56% of the jurisdictions, an increase of 26%. And I have collected 23 clinical case publications of individuals acquiring or becoming sick with aspergillosis affiliated with cannabis use. So, yeah, I have a very short talk. So where might we go from here? The 6 specific pathogens that are required in the California model are not all the human pathogens in cannabis flowers. I'll be the first to admit that. So how do we identify these additional human pathogens, which would lead to a possible creation of a consensus set of microbial testing rules to be considered by the federal government? And so this research study is a massive study. It's more in my brain. So this would be a national cannabis flower microbiome study. So every legal jurisdiction that has legal cannabis, one would determine a statistically significant number of cannabis flowers, which is a minimum of 400, then they would be sampled in the different regions of the jurisdiction, noting what type of cultivation was used, outdoor, indoor, hydroponics, so forth and so on, and the actual technology has already been developed. So one does a direct DNA extraction from the flower. The second step is the amplification of that DNA for both the bacteria, the yeast, and the fungi. So the sequences that you amplify for the bacteria are the 16S ribosomal genes, and for the yeast and molds, it's called the ITS sequences. So one has all this DNA now that you've amplified, and then you do sequencing of this DNA, and there's now nanopore technology developed by Oxford, the Oxford Technology Company, where the sequencer is a little bit bigger than a flash drive, and the computer is quite little, and you can buy one and play with it for $6,000. And then you have to contract or have in-house a bioinformatics expert, and believe it or not, and we've already done this at MGC, is that you can identify every genus and species in that cannabis flower. And also you can determine the relative amount. So now you have all this massive information. It's a lot. So what do you do with it? And I don't have the full answer of the composition of the stakeholders group. I don't. But I know that we need infectious disease doctors. I know we need to have clinical microbiologists and also risk assessment experts. Just because you have a human pathogen in cannabis flowers, if it's at a super low amount, does not necessarily mean that you have to add it to the list. But if you find or identify human pathogens at high levels in number of flowers throughout the country, then you want to keep it in the list. And then literature search experts are very, very important because they can do massive literature searches to see what are the microbial species that are human pathogens, which ones are opportunistic pathogens, and so forth and so on. And then through just dialogue, they identification of additional subject matter experts that could be included in this dialogue. And from all this big data set, one can then determine a consensus set for microbial testing rules for a state, for a region of the United States, or the whole country. I don't know how this is going to come out. And I hope to see, I'm sharing this slide simply to plant the seed for people to consider from now into the future, and hopefully this study will be conducted in a stepwise manner until we cover the entire country. And I'd like to acknowledge the individuals who helped me in this massive project: Dhanasri Nair, Dhanasri Pawar, and Shravani Chobhi, who updated the database and created all the data sets, and Summer Hall helped me visualize some of the data. In this last slide, I have my email up there if you want to ask me any questions. Email me to continue this dialogue, and at this time I'd like to entertain any questions. Thank you. And I kept my talk short.
Hi, Sherman.
Hey, Jeff.
I never dreamed anyone can make a talk about regulations so exciting. It was really great. I wanted to ask you, I've talked to a couple of people with history working in the USDA, and they have told me about a lot of, institutional knowledge about agricultural pathogens. Is there still information that's kind of known from other crops that we're missing here that we could use to kind of have a better guess of what to look for?
Well, the thing is that you mentioned the USDA, and I worked in public health and environmental chemistry, and in our laboratory in New Jersey, we had some interaction with the Department of Agriculture, and again, thank you very much for pointing that out to me. I really have not had the opportunity to ask them in terms of, I know that they focus on the plant pathogens, but with all the information and all the experience they've had, I'm sure they've identified some human pathogens, so I think it would be a good source of information.
Yeah, like you should look into like berry sorter's lung and rose grower's thumb. There are a bunch of them that are known, especially from agricultural occupation, and there is quite a lot of information out there.
Right, okay, we'll talk about that, thank you. Is there any other questions? Thank you very much for listening, thank you.